Alright, welcome to today's lecture. Today's lecture we're going to be talking about central dogma and DNA, focusing on two big questions: what is DNA and how is the genetic code expressed as a phenotype? Phenotype, you might not know what that word means. Phenotype has to do with your physical appearance. So how does your genetic code determine who you are? That's the question we're going to be trying to explore today, building off some of the things you might have learned in 7th grade. So first off, what is DNA? DNA is a double helix. It's made of nucleotides. It's tightly bound as chromosomes in the nucleus. You might also hear of something called histones. Histones are proteins that help with the binding of DNA. They're proteins that help with the binding of the DNA, and that helps it to be stored really tightly. We also know a little bit about DNA when we built our model, that there are base pair rules, meaning that some things always go with other things. DNA is made of nucleotides. There are 4 nucleotides, A, T, C, and G, as they are affectionately abbreviated as. Those stand for adenine. Adenine always goes with thymine, they're best friends. And cytosine always goes with guanine. And as a reminder, what is a nucleotide? A nucleotide has to do with the fact that it is made up of a sugar, a base, and a phosphate backbone. And that allows it to have something on the outside that's hydrophilic and something that's hydrophobic on the inside. So water liking and water hating. And that has to do with kind of DNA and how it binds and moves around things. So now we talked about what DNA is, we're going to talk a little bit about how DNA is expressed. DNA is expressed in something called the central dogma. The central dogma was an idea that describes how DNA is transformed and translated, an idea that describes how DNA is turned into a genetic footprint or a genetic plan. So in this plan, we go from DNA, that DNA is replicated, so talk a little bit about replication in class. DNA then becomes something called RNA, and there are 3 types of RNA: messenger RNA, transfer RNA, and ribosomal RNA. And this is through a process called transcription, where DNA from inside the body, inside the nucleus, is able to go out into the cytoplasm, and then from RNA we're then able to go to a protein, which again was one of the 4 macromolecules. Proteins are then able to function in the body, such as enzymes, and they can also provide things like structure, such as muscles and cartilage and that type of thing, where it can be used as a building structure. So the idea is that we have DNA goes to RNA, goes to protein, and that happens through the process of transcription and translation. So now a little bit more depth into each of these. In transcription, who are the key players? In transcription, the key players are DNA and an enzyme called RNA polymerase. Poly meaning many, polymerase meaning to build. So we have something that builds messenger RNA. And other than that, those are kind of the big key players. There's also kind of a pore in the nucleus that allows the stuff to come out. So we're inside the DNA, and kind of the first step that needs to happen is DNA unwinds partially. It's a double helix, so it needs to unwind to allow things to have access to the DNA code. RNA polymerase attaches, and makes messenger RNA. So what would be an example of that? If we had a DNA strand that was TAC, CGA, one difference between mRNA and DNA is that RNA has something called uracil. So uracil replaces thymine. There's no thymine in RNA. So then we have our complementary strand of RNA, This would be the template, and this would be our new mRNA. You'll learn a little bit about how they're anti-parallel, but just to simplify for today, A would go with T, U would go with A because there's no more thymine, G would go with C, G would go with C, C would go with G, U would go with A. And that would be the start of your new messenger RNA that would then get made and sent out to the cytoplasm. So now that we have this code, this messenger RNA floating around, so now we have messenger RNA in the nucleus. What happens next to build our protein? We're looking at these key players. We're looking at the messenger RNA. And we're looking at something called a ribosome, which kind of looks like a Pac-Man type thing that puts everything together. We have something called tRNA, which is called transfer RNA, that brings things to the message. And lastly, we have, how would you describe it? The amino acids that are used to build the protein, and those are attached to the tRNAs. So tRNAs kind of look like keys or little clovers, and the amino acids kind of sit on top, because as the amino acids are built, it's made up of amino acids, which I'll abbreviate as AA. So now we talked about the key players. Let's hit on kind of the steps. So the first step is the mRNA finds the ribosome. The ribosome has a set of codes. It reads the mRNA in sets of 3 called a codon. An example of a codon would be something like AUG, would code for methionine. And we'll practice this in class. But the idea is you always want sets of 3 that will code for one amino acid. From there, it'll continue on and on and on, and just like before, eventually it will reach the end of the messenger RNA, and a new protein is released. If you're interested in this kind of stuff, this is the kind of topics you would hit on in AP Bio. The idea is that the ribosome reads the mRNA and then leads to a new protein being released that can then affect your phenotype. So kind of what are the results of gene expression? We know that you can have different DNA but similar phenotype. This has to do with the genetic code. And you'll see a big table, and that table will show you that you can have different DNA. For example, different things will code for the same end codon or a stop codon, but have 3 different letters. Lots of things can go wrong in this. There's a lot of steps and there's a lot of moving parts, and that's a mutation. A mutation is a change in the DNA. And just a reminder that a change in the DNA, it can be good, it could be bad, or it could have no effect. A lot of people like to think of DNA and mutations as something horribly bad, leads to cancer, or they might think of it like Teenage Mutant Ninja Turtles, something that's amazingly good. But the result of this central dogma is that you can have a lot of variety, and there's a lot of details and factors involved to making a functional individual. So to wrap up on our last slide, just wanted to make sure you got the summary of what's happening. We know that DNA is the instructions of life. It's important to know its structure and how it acts as the instructions for life, and how it change in the DNA in the beginning can later affect DNA later. We also want to recall what the central dogma is, and how your genetics can affect how you look and behave or disease states, and because of that, that's because of the proteins. So keeping that in mind, proteins are kind of the center of all this because it affects your phenotype and what you'll look like and be. So if you have any questions about central dogma, feel free to shoot an email or give me a call. Be more than happy to help you out. So otherwise, have a great day, and I look forward to talking more about the stuff with you in class. Have a good one.